Hello students, let's start with new topic of 11th standard today, that is mitosis. Okay, in previous lecture we have studied about cell division. Okay, that cell division means when a single cell gives rise to two or more daughter cell, that is called a cell division. Then we have studied cell cycle. Cell cycle is completed in two phases, that is interphase and M phase. So M phase we are going to study now in this lecture. M phase means where stands for mitosis or it stands for meiosis. Okay, so here we are studying first mitosis. Now see, what is mitosis? Mitosis is a type of cell division. Okay, mitosis is a type of cell division in which a single cell divides or it gives rise to two daughter cell. Okay, mitosis. What is mitosis? Mitosis is a type of cell division in which a single cell gives rise to two daughter cell. This mitosis is also called as equational division. Equational division. Why it is called as equational division? Because the number of chromosome present in the mother cell and daughter cells are equal. For example, if the mother cell is having 46 chromosome, the daughter cell will also have 46 chromosome. Means the chromosome number is maintained in mother and in daughter cell. Okay. So what is mitosis? Mitosis is a type of cell division in which a single cell gives rise to two daughter cell. Mitosis is also called as equational division because the number of chromosome is maintained in daughter and in mother cell. Next, now mitosis is completed in two phases. First one is karyokinesis and another one is cytokinesis. See, karyon means nucleus and kinesis means division. In the same way, cyto means cytoplasm and kinesis means division. So what is the meaning of karyokinesis? Karyokinesis means division of nucleus and cytokinesis means division of cytoplasm. So what we are studying here? We are studying about mitosis. Mitosis is completed in two phases, that is karyokinesis and cytokinesis. Karyokinesis is division of nucleus and cytokinesis is division of cytoplasm. So let's start with karyokinesis. Karyokinesis is again completed in four subphases: prophase, metaphase, anaphase, telophase. Okay, now before proceeding with prophase, metaphase and anaphase and telophase, we will see what is actually happening in mitosis. See, what is mitosis? Mitosis is nothing but it is a type of cell division in which a single cell gives rise to two daughter cell. See, mitosis means it is when a single cell gives rise to two daughter cell, that is called as mitosis. Okay, mitosis is also called as equational division. Why it is called as equational division? Because the number of chromosome in mother cell and in daughter cells are equal. If the mother is having 46 chromosome, the daughters will also have 46 chromosome. Okay, so now we are studying about mitosis. Mitosis is completed in two phases. First phase is called as karyokinesis and another phase is called as cytokinesis, where karyon means nucleus and kinesis means division, cyto means cytoplasm and kinesis means division. So mitosis is completed in two phases, karyokinesis and cytokinesis. Now this karyokinesis is again completed in four subphases. How you can keep this in mind, these four phases? First one is P for prophase, second one is M for metaphase, another one is A for anaphase, okay, and last one is T for telophase. In this way, how you can keep this in mind? PM at. You can keep in mind PM, that is Prime Minister at Pune. Okay, PM at, where P stands for prophase, M stands for metaphase, A stands for anaphase and T stands for telophase. Okay, so what we have studied here, we are studying mitosis. Mitosis is a type of cell division in which a single cell divides or give rise to two daughter cells. Mitosis is also called as equational division because why it is called as equational division? Because the number of chromosome in mother cell and in daughter cell remains same. Then what we have studied, mitosis is completed in two phases, karyokinesis and cytokinesis. Mitosis is completed in two phases, karyokinesis and cytokinesis. Okay, karyon means nucleus, kinesis means division. Cyto means cytoplasm. Kinesis means division. So what is the meaning of karyokinesis? It is nothing but division of nucleus. What is cytokinesis? It is nothing but division of cytoplasm. Now karyokinesis is again completed in four phases. PMAT, PM at, where P stands for prophase, M for metaphase, A for anaphase and T for telophase. So let's start with prophase. See what things are happening in prophase. In prophase, the chromatin network, this is nothing but chromatin network. Now what happens in prophase? The chromatin network get condensed. Why the condensation is taking place? Because this chromatin network have to get this chromatin network is going to convert into chromosome, and then only the division of chromosome will take place. Okay, so first of all, what is chromatin network? You should know. Chromatin network, it is nothing but it is the network of protein molecules and DNA molecule, okay, which is duplicated or which is formed in S phase of interphase, and they are intertwined with each other, and the protein and the DNA which is duplicated and intertwined, they are mixed with each other. Okay, so that form is called as chromatin network. And see what happens in early prophase. In early prophase, the chromatin network, now chromatin network is clear to you. Chromatin network is nothing but it is the network of protein and DNA which is duplicated in S phase. So that is called as chromatin network. So here what happens in early prophase, chromatin network get condensed to form chromosome first. Then second, what is happening? Nucleolus. Can you see this part? What is the function of nucleolus? It is a dense body present inside the nucleus. Nucleolus is responsible for the formation of protein. Okay, so nucleolus is there. Now see, protein is not required. Already in interphase, protein synthesis took place. So now nucleolus is not required. So nucleolus will start disappearing. Now, nuclear membrane. This membrane of nucleus is called as nuclear membrane. This also we don't require because afterward here fibers are going to form, and because of this membrane disturbance will be there. No? So this nuclear membrane will also start disappearing. Nucleolus starts disappearing. This nuclear membrane also starts disappearing. Then many here, if cell is there means many cell organelles will be there. Membrane bound cell organelles, for example endoplasmic reticulum will be there, Golgi apparatus will be there. All the membrane bound in organelles, cell organelles will also start disappearing, because whenever there will be the formation of spindle fibers, no disturbance of cell organelle should be there. So all these things, that is nucleolus, nuclear membrane, membrane bound organelles like endoplasmic reticulum, Golgi apparatus, they start disappearing. Okay, then what happens? See, this is centrosome. This centrosome which is already duplicated in S phase. Okay, in animal cells centrosomes are present, and this duplication took place in N phase, S phase of interphase only. Here in early prophase, the centrosome divides. Divided centrosome start migrating towards the opposite pole. Division already took place in S phase of interphase, and the divided centrosome, duplicated centrosome, they start moving towards or migrating towards the opposite pole. of the cell. Okay, then what happens in late prophase? This thing is happening in early prophase. What things are happening here? Three things only you have to keep in mind. Chromatin network get condensed to form chromosome, nucleolus, nuclear membrane, and membrane bound cell organelles like endoplasmic reticulum, Golgi apparatus, they start disappearing. Third point, the divided or the duplicated or the separated centrosome start migrating towards the opposite pole of the cell. Now, what happens in late prophase? Early prophase means starting of prophase, and late prophase means what happens exactly at the end of the prophase. Now, what happens in late prophase? Due to condensation of chromosome, due to condensation of chromatin network, what happens? Chromosomes are visible over here. If they are observed under the microscope, no, here what type of structure is visible? Two sister chromatids and one centromere. Means because of the condensation of chromatin network, chromosome structure formation or the condensation of the chromosome takes place. They are not becoming too much thick over here, but as compared to early prophase, they forms little bit thickness or thick structure. Okay, so this is all about prophase, where we have completed early prophase and late prophase. Now let's start with metaphase. Now see how you can keep this in mind. M for metaphase, M for middle. Okay, in this way you can keep in your mind. Second phase is metaphase, where M stands for metaphase and M stands for middle. Okay, now see what happens here. Here, no, chromosomes which are completely condensed now, they become very short and thick. Okay, they become very short and thick, and the sister chromatids and the centromere, these are the sister chromatid, and this center, this one is called as centromere, these are called as sister chromatid. Okay, now this chromosome becomes short and thick, and the sister chromatids and the centromere becomes very prominent. They are easily visible here. Okay, what is happening in metaphase? First thing, the chromosomes becomes short and thick. They undergo coiling. Okay, and the structure becomes very short and thick and prominent also. Prominent means these two chromatids, that is sister chromatids and centromere, is clearly visible or it becomes prominent. Then what happens? All the chromosomes. Now see again what is happened over here. Nucleolus completely disappear, nuclear membrane completely disappear. Now what happens? The chromosomes arrange themselves, or all the chromosomes lie on the equator, or you can say on the equatorial plate, or you can say it is called as a metaphase plate also. What is happening in metaphase? All the chromosomes lie on the metaphase plate or on the equatorial plane. Okay, then what happens with the centrosome? This total full structure is called a centrosome, and inside the centrosome the centrioles are there. Okay, centrosomes which reach to the opposite pole of the cell, they give rise to two types of spindle fibers. What is spindle fibers? Spindle fibers are nothing but these are the fibers made up of protein, and that protein is called as tubulin. So in metaphase, there is formation of two types of fibers. Can you see one fiber? The fiber which is arising from centrosome and coming up to the centromere of the chromosome. This fiber is called as chromosomal fiber. And the another fiber which arises from one centriole or centrosome to another, that is from one pole to another, that is called as interpolar chromosomal fiber or interpolar fiber. Okay, so again I'll repeat: M phase, M for meta, M for middle. Okay, what happens here? Here also three things are happening. Okay, first one is here chromosome. What happens with the chromosome? Chromosome becomes short and thick. Chromatin and centromere is clearly visible or it is prominent, and chromosomes arrange themselves or they get lie down on the equatorial plane or on the metaphase plate. Okay, this has happened with chromosome. What about this other membrane, say nuclear membrane and nucleolus? Nucleolus, nuclear membrane, Golgi apparatus, and endoplasmic reticulum, everything completely disappears. What happens with the centrosome? Centrosome reaches to the opposite pole and they give rise to two types of fiber. The fiber which arises from centrosome and reaching up to the chromosome is called as chromosomal fiber. And the another fiber which is arising from one pole and going up to the another pole, that is called as interpolar spindle fiber. So here we have completed metaphase. Let's start with anaphase. What happens in anaphase? In anaphase, see, this is chromosome. Okay, this is chromosome, means these are the chromatids, and this is centromere. What happens in anaphase? In anaphase, the centromere divides and results into the formation of daughter chromosome. Means from one chromosome, total two chromosomes, daughter chromosomes are formed. Okay, then what happens? The daughter chromosomes are pulled in another or in opposite direction because of this chromosomal, because of the contraction of chromosomal fibers. The daughter chromosomes, 46 daughter chromosomes are pulled towards one pole, remaining 46 chromosomes get pulled towards another pole. Again, I am repeating what is happening over here. In anaphase, the centromere of the chromosome, each and every chromosome centromere divides. Okay, what is happening in anaphase? Centromere divides, okay, which results into the formation of daughter chromosome. Okay, so what happens actually? There are 46 chromosome, and from 46 chromosome, 92 daughter chromosomes are formed. Out of this 92 chromosomes, because of the continuous contraction of chromosomal fibers, 46 daughter chromosomes are pulled towards one pole and 46 are pulled towards opposite pole. Okay, actually this pulling of chromosome is how they are pulling. There, here near to the centrosome, no, one small disc shaped structures are there, that is called as kinetochores. Just near to the centromere, one small disc-like structures are present. That is called as kinetochores. These kinetochores, no, actually the chromosomal fiber get attached to the kinetochores and the daughter chromosome get pulled. Okay, so half number of daughter chromosome get gathered towards one pole and remaining half number of chromosome gathered towards the opposite pole. So this is happening in anaphase. Now last stage is telophase. Now see what happens in telophase. In telophase, see the daughter chromosome get gathered on the opposite pole. 46 daughter chromosome at the one pole, 46 chromosome towards the another pole. Now this telophase is just opposite of prophase. In prophase, what had happened? Nucleolus completely disappear. Here, nucleolus reappear. There in prophase, nuclear membrane disappear. Okay, here the nuclear membrane again reappears around the daughter chromosome at both the poles. Here at one pole, daughter chromosomes are gathered, 46 daughter chromosome. Here also at the another side or another pole, 46 daughter chromosomes are gathered. So what happens? Around this daughter chromosome, nuclear membrane reappears on both the around both the daughter chromosome or around both the group of the daughter chromosome. Here towards one group, nuclear membrane reappears. Here towards opposite group, again one more nuclear membrane reappears. Inside the nuclear membrane, there is formation of nucleolus, which disappears during prophase, is reappearing in telophase. Okay, then what happens here? Now see, now there is no use of centrosome. No use of centrosome, no use of spindle fibers. So what happens? The spindle fibers break down over here because the purpose of spindle fiber is over. Okay, so spindle fiber break down and it is absorbed in the cytoplasm. In other way, you can say spindle fibers completely disappear. Okay, so this is all about telophase. What things are taking place here? In telophase, the daughter chromosome which are gathered here at the opposite pole, around those daughter chromosome, nuclear membrane formation took place. Inside the nuclear membrane, means two nuclei are formed here. Okay, and inside the nucleus, inside each and every nucleus, nucleolus formation took place. So from one nuclei here in telophase, from one nucleus here in telophase, two nuclei are formed. Okay, so this is all about telophase. Now, so this is all about telophase. Now what happens in cytokinesis? Okay, cytokinesis is nothing but division of cytoplasm. Cyto means cytoplasm and kinesis means division. So what happens in cytokinesis? In animal cell and in plant cell, the division is taking place. Division of cytoplasm is taking place in different way. Okay, so what exactly happens in animal cell? In animal cell, the furrowing takes place. This is the cell wall. Here what happens? This cell wall furrowing or notch development takes place. This notch continues, okay, from periphery towards the center. This notch development or the furrow development takes place from periphery, from outside towards the center, and finally what happens? It results into the separation of two daughter cell. Means this is one cell and this is another cell. One more thing is happening here. When the nuclei is forming, no, these chromosomes, daughter chromosomes again get uncoiled, and again there is formation of chromatin network. Okay, and then whenever there is the formation of chromatin network, later on both the cells get divided. In animal cell, furrowing takes place. Furrow, this outer side membrane notch development takes place from periphery towards the center, and both the cells get separated from each other. While in case of plant cell, no furrowing is taking place in cell wall. What is happening here? Here at the center, in plant cell, at the center, hard cell plate formation takes place at the center, which proceed towards the outer side. Here from the center it is proceeding towards the outer side or towards the periphery, and finally both the cells get separated from each other. Okay, which, and afterward this cell plate get converted into middle lamella, which is present between the two adjacent cell. So in this way we have completed all about mitosis.